Good afternoon, everyone. My name is Jason Roberts. I'm from the University of Queensland, the Royal Brisbane and Women's Hospital, and I'd like to welcome you to the Antimicrobial Susceptibility Testing and European Committee on Antimicrobial Susceptibility Testing, so that's the EUCAST expert update. So we have a true international speaking cohort today who are leaders in their own right for the topics that they're talking on, and we're very grateful to have each of the speakers. Now, the first speaker is Professor Jean-Philippe Lavigne, who comes from CHU Nîmes, so that's a university hospital in Nîmes, which is in southern France. Now, I can't see at the moment that Professor Lavigne has joined, and so I'm just waiting for him to say, bonjour, Jason, I have joined. And so, Professor Lavigne, please feel free to just acknowledge that you are online, and then as soon as you join and you speak up, then we are able to then commence. So, Professor Lavigne, are you online? So we might just wait a couple of moments until Professor Lavigne joins. I'll come back to you again soon.
Oh yeah, it's Jean-Philippe Lavigne. You said, do you hear me? No, just in the chat.
Just above Crispin's, a bit further down.
Professor Lavigne, we can't hear you at the moment.
Can you make him a panelist? Oh, John's there too.
Professor Lavigne, can you hear us now?
Yeah, I hear you.
Ah, perfect. Lovely.
You hear me?
Yeah.
You hear me? You hear me, Jason?
Yes, we can. I'm just making sure that you're able to share your screens. Yeah, screen, yes, that should be. So, welcome, Professor Lavigne. We have about 100 attendees online at the moment from various parts of Australia, and I'm sure from other countries as well. We're very much looking forward to hearing your presentation on susceptibility testing, which would be from a European perspective. So, by way of introduction, Professor Jean-Philippe Lavigne is the head of the Department of Microbiology and Hospital Hygiene, as I said, at Nîmes University Hospital, and he also has an affiliation at Montpellier University. He is an expert in communicable diseases, and I've been very fortunate enough to meet Professor Lavigne and have also heard him speak at a symposium that our Centre of Research Excellence, REDUCE, held at CHU Nîmes in 2019 as well. So it's a pleasure to welcome Professor Lavigne to give his presentation, and I invite him to share his screen and commence. Thank you, Jean-Philippe.
Thank you, Jason. Have a problem to open my presentation?
Try now, please.
Yes. Now, okay.
We can see your screen now, so.
It's okay? It's okay for you, Jason? I can begin?
Yes, please. Thank you.
Thank you so much. So I'd like to thank the organization of this webinar, and particularly Jason, to invite me to present some advancing antimicrobial testing. Yeah. Firstly, I declare that I have no link and no conflict of interest with any companies sit in this presentation. So when a microbiologist performs antibiogram, he expects to obtain a response on the susceptibility of the bacterial strain isolated from a sample. And on the other side, the clinician are waiting to obtain this result anxiously because time is very important in best management of patients. So we use different analytical methods to test the susceptibility of the strain against a panel of known antibiotics. And the most used techniques are based on bacterial culture. And we can evaluate two main indicators: the minimum inhibitor concentration, the MIC, by microdilution or e-test, and the different diameters of inhibition zone by disk diffusion assay. And a standardization of the results was now proposed according to international guidelines criteria of EUCAST in Europe, or CLSA for the rest of the world. And the antibiogram data does not only inform the team of one individual patient, but altogether the collected antibiogram reports can be aggregated into one hospital, a network, or a region, a country, and around the world. However, to obtain an antibiogram, the clinician have to wait a long time, around 48 hours. And one of the main objectives is to decrease this time. And I propose you this morning this morning in France to present the different advances in the field. And firstly, I present you the phenotypic approaches. So among these phenotypic methods, you have the fully automated susceptibility testing system. In the lab, you just prepare bacterial suspension, and 6 to 11 hours after, you have the result and is interpretation. The machine gives frequently approximately the MSI value, not totally adapted for therapeutic adjustment. Some new evolution are available very recently for the VTEC2 cards. It's a machine built by Biomérieux in France, and you have now in this new evolution a larger panel of antibiotics against gram-positive and gram-negative bacteria, more with recent antibiotic. For example, for gram-negative bacteria, you have now ceftolozane tazobactam, ceftazidime avibactam. Moreover, the MIC range are also larger, given more accurate MIC value. But the main problem is this solution was effective after 48 hours to obtain the first antibiogram. So it's very long time before the use of these cards. The EUCAST group has proposed new requirement with the determination of MIC, some MIC in the liquid media, and to perform this experiment, you have the unique solution, and with this solution you can perform MIC value to vancomycin, teicoplanin, colistin, or tazosaline. And this solution exists, is very interesting, but also this solution is very long to obtain, corresponding to an overnight time. Other solutions, the immunodetection tools are also available. On your right, you have Adelaide PBP2A. This solution can identify MRSA in less than 10 minutes on culture colonies, but also directly from blood culture. And this tool is very adapted to detect MEC-A genes, but this solution didn't detect MEC-C gene. You have the Corisbio concept. Corisbio concept is a Belgian company, and this company has launched different tests for the detection of carbapenemase by immunochromatography. And here you have the Resist 5.0 test, and it is the most extended panel to detect carbapenemase producing the enterobacteria on culture colonies with excellent performance, approximately 100% of sensibility and specificity. And finally, you have the beta-lacta test, and this test detects the presence of class A beta-lactamases directly from blood culture or urines, allowing to rapid best use of antibiotics. You have a very recent and interesting evolution to obtain a phenotypic response is a French company, IQA. This society has launched a machine with automation of incubation, reading, and interpretation of antibiogram performed on disk diffusion method. They use a new Gelozago MHRC, and with this machine you can obtain results after 70 hours for blood culture and 40 hours for urine, earlier than the result obtained with a conventional antibiogram. Moreover, the performance of this test is very high, more than 97% of categorical agreement for Enterobacteria or Staphylococcus aureus. And the interest of this solution is to observe, detect all the resistance mechanism, and is very reproducible solution. So in conclusion to the phenotypic solution, you can see that this technology are highly sensitive, rather definitive in profiling antibiotic susceptibility. You have the determination of all resistance mechanism with the last solution. The low cost is very important, but the result is slow, very slow to obtain. You have a unitary test. There is also all these methods are inefficient to detect non-culturable or slowly growing bacteria. The second approach to reduce the delay of antibiogram to obtain antibiogram is molecular diagnostic tools. So here you have many solutions, and all the molecular base test, all this solution provide firstly bacterial identification, and secondly a detection of resistance traits directly on the patient samples. So it's very interesting, and you have two main trends, two main approach. The detection based on molecular markers that confer resistance, and the syndromic approach. So among the numerous solutions, we could present the point of care system. It's a GenExpert built by Cepheid company. It's a very old technique, but always interesting for the clinical lab, clinical microbiology lab. This system, in fact, is an equivalent of PCR from dummies. This solution detects some resistance genes directly from the samples, and these tests have excellent performance, more than 90% of sensibility and specificity, and it's very rapid. The result could be obtained in approximately 1 hour. However, the panel are limited. You detect MAC-A, some carbapenemase, and Van-A in vancomycin resistant enterococcus, and also resistance in Mycobacterium tuberculosis. So the panel is very limited, and another problem is the cost. This solution is particularly expensive. In France, one test is around 40 euros. The syndromic approach is a very exciting solution. This solution allows to screen a large panel of samples: blood culture, respiratory tract, digestive, CSF, urine, etc. And the screening was performed with a great potential of target. And you have many solutions and machine actually or currently. The main limitation of this solution are the technical time needed to perform PCR, and some solutions are not totally adapted for routine in a microbiology lab. You have also a different fast syndromic approach, and this approach are also available with a turnaround time of about 1 hour. This solution proposes a different panel for bacterial identification and detection of K resistance marker directly from a sample. However, the solution remain limited from sample, mainly from blood culture, and the resistance panel is also limited, MAC-A, Van-A-B, or KPC. And the most important problem with this approach is the cost, very expensive. In France, the cost of one reaction is more than 100 euro. So in conclusion, the PCR solution is very interesting method with a very short turnaround time. This solution can detect a non-culturable bacteria. You have this potential of a multiplexable solution. Is very interesting, but often this require sample processing steps to purify and amplify target genes. The presence of resistance marker may not correlate with phenotypic resistance. You could have some mutation in some resistant gene, and the solution could not detect this mutation. You have also a limited set of pre-identified genes. For example, the main gap for this solution is the no detection of AMC derepression and the potential resistance to third generation cephalosporin. The cost is also a big problem to this solution, and some solutions have a limited sensitivity, notably when you work directly on samples. The third approach to reduce delay to obtain antibiogram use mass spectrometry. So mass spectrometry is a revolution in bacteriology. This method is based on the rapid ionization of the bacterial protein using a laser pulse directly from a culture colony or cell pellets from the clinical samples. The calculated mass of the ion is a specific sample fingerprint of the bacterial species, and this technique is nowadays widely used in the clinical microbiology labs. This technique requires fresh colony, the better use of this solution, but also you could work on the blood culture after sample preparation. The workflow is very high. You can screen 16 to 384 sample in a same template, and the analysis is very fast. One result for identification is obtained in 30 seconds, and to detect resistance is around 2-3 hours. So to detect resistance, for example, to detect carbapenemase activity in gram-negative bacilli with the Malditov, you incubate the bacterial samples with an antibiotic. Here you use ertapenem, and analysis through Malditov detect different mass peaks obtained at the beginning, before the incubation and after incubation. And here you can see if you have the hydrolysis of beta-lactam ring by the carbapenemase, yes or no. And you can conclude on the presence of absence of this carbapenemase. Some different applications are now available. Firstly, you have the detection of MRSA. In literature, this detection is contradictory, and all the team are not okay with this method, but you can find some presence absence of specific peaks of MRSA or MSSA strain. You have also the detection of ESBL by enzymatic method, and this method is equivalent with the method present for the carbapenemase detection. With MALDI-TOF, you can find also fluoroquinolones resistant Streptococcus pneumoniae, and you can find also resistance in the fungi, for example, Candida. So MALDI-TOF is very interesting because the result is very fast. You have a very low cost of a sample. The analysis is under €1, and you can cover a very broad range of species. The main problem is the cost of the machine. One of the machines are relatively bulky, and this solution may not provide a direct correlation between the presence of hydrolytic enzyme and the phenotype of resistance. For example, with this method, you can't detect efflux or impermeability. So you can detect carbapenem resistant enterobacteria with this solution. Different other solutions are now available, and I propose to present this other solution. Firstly, you have different imaging-based technology developed at this moment, and this solution can provide the identification of bacteria, but also perform or provide antibiotic susceptibility testing. This solution represents an impressive emerging technology in the field of clinical microbiology. Here, for example, you have the oscilloscope system. This system is one of these solutions based on real-time analysis using an automated optical detection system. The average time to obtain the results is around 1 to 4 hours, and this system have been developed to detect the susceptibility of E. coli in monoculture, and the solution has been evaluated after blood culture. The second solution is an accelerate solution. This machine uses a multi-channel test cassette that allows the direct observation and measurement of bacterial growth in a real-time approach. Direct measurement of bacterial growth or death is obtained from for a panel of antibiotics, and different algorithms convert the growth curves into MIC values. And the results are provided in approximately less than 7 hours. So we have tested and evaluated this solution in our lab. This solution is very, very interesting method, but the cost of this cassette is too expensive to be used in routine labs. In France, this solution is around 300 € for one blood culture bottle, so it's impossible to use in routine. Another solution here is a bacterial scan. This bacterial scan is an electro-optical based technology using laser light scattering. It measures the angular variation in the intensity of light scattered by a laser beam that pass through a bacterial sample. The angular variation depends on the number and size of bacterial cells in suspension, allowing to detect very low value of optical density and to measure bacterial growth. Multiple measurements every 3 minutes are done, and to capture the signal, you have a two-dimensional camera sensor, and this you can generate a density value, and the instrument perform reliably down to minimum density of 10 CFU per milliliter. So this solution is very adapted in urine analysis, but one of the main problems is this solution is interesting in monomicrobial infection. And we know that many contamination could be observed with urine sample. So it's a main problem with this solution. And this bacterial scan can process 16 samples simultaneously. It was evaluated using E. coli, Staphylococcus aureus, and Pseudomonas aeruginosa strain, and a very close agreement was observed with a conventional test. And the overall time of the essay was approximately less than 10 hours. The last solution, this solution is based on the single-cell morphological analysis, and this solution uses a bright field microscopy to determine antibiotic induced change in cells immobilized on an agarose chapel chip. This solution is now commercialized it's a multi-path platform, and this platform applies non-magnified digital imaging for the detection of biomolecules tagged with antibody-coat fluorescent nanoparticles. And antibody coat magnetic particles are used to bind to target cells and to pull them down to the camera surface. The system counts individual targets in large area and perform growth monitoring for multiple target cells, and enables to give and to determine MIC values. The system is developed for 40 antimicrobial susceptibility tests, and the overall time of the assay vary between 2 hours to 8 hours. Evaluation are needed now to clearly evaluate the potential of this platform. So in conclusion, with this imaging-based solution, with advantage, you have a shorter turnaround time. I'm enabled for directly monitoring bacterial growth in sample and detection of MIC values. But initial culture step is still required. So you have a minimum on 1 day before this analysis. Complex algorithms are required for accurate bacterial phenotype analysis. I think publications are needed to confirm the interest of all this solution. And the cost, we have no clear idea of the cost of this solution. Finally, I present you other technology. Among this technology, you have the life-scale instrument. And this instrument is use a correlation between cantilever vibration and biomass to detect MIC values. Each frequency shift reflects the mass measurement of an individual microbe, and culture are sampled at a regular time interval to measure growth in microbe mass and number with time. And you perform the measure before and after exposition to antibiotics. To measure a microbe, the resonance frequency is monitored as the microbe passes through the microchannel resonator, and the system performs automated cell-counted mass measurement and visual observation of liquid sample in the aim to detect MIC value. And this solution is very fast, simple, and complete automation. Another solution is colorimetric sensor arrays that provide an inexpensive method to detect volatile organic compounds, VOCs, associated with microbial metabolism. And this technology e-nose, electronic nose, and this technology use a selective sensor of small molecules that are sensitive to variation of the VOCs. This system can detect an electronic aroma signature to identify bacteria and discriminate, for example, MRSA and MSSA. The different VOCs produced by these two bacteria different. And the overall time of the assay is around 4 hours. You have a determination of MIC. This solution is just yet developed in a European country, and no evaluation, clinical evaluation have been provided to date. A last solution is a microfluidic approaches. Resistance profile are determined based on the electrochemical reduction of a redox active molecule, the resazoline. Using this approach, you can detect bacteria at concentration of 1 CFU or above, and it exhibits a similar MIC profile for the antibiotics they tested when compared with a conventional culture-based method. And all this approach are obtained in approximately 7 hours. In the same way than other solution, evaluation are needed to clearly evaluate the potential of this solution. So to conclude with this other solution, you can see single cell sensitivity, rapidity to obtain results, but often the solution requires pre-enrichment of bacteria, sample processing step to obtain single cells prior to measurement, and there is a limited throughput. Finally, in the future, many other solutions have been developed using very different technology. You can see electronical sensors, synthetic biology approach, notably using bacteriophages. Perhaps we could use some host markers. You have NMR spectroscopy, molecular technology, notably using LAMP technique, mass spectrometry, and also sequencing technology. And I would like to finish this this presentation with perhaps the future using the precision medicine. You can see here a Minion. It's a point of care sequencing system, very small and portable solution. And the DNA being analyzed are sequenced in this solution instantaneously, and the sequence readout are aligned to a gene profile database in parallel, and you can obtain a result very quickly of all the genome of bacteria. So you can identify species, but you can also observe all the mutation or the discover of resistant gene in a bacteria. The required bioinformatics is challenging. I think it's one of the main evolution of microbiology labs, and universal open database are needed to interpret the results. So as their properties and application area differ, all the system present at this moment may have a good feature. Rapid growth-based diagnostic system rely on accelerated cultivation. For this reason, it's necessary to validate every new technology carefully against the standard test accepted by EUCAST or CLSA, and the MIC breakpoint must be redefined for each system and each tested organism. This requirement definitively retards the development of this technology. So many thanks for this presentation, Ernie. To conclude, the last conclusion, Jason, I have a link with Australia. You don't know, but I have a link in Australia. I just see you. This link is my dog. He's an Australian Shepherd, and it's very important to tell you that Australia is in my house.
Well, that makes a lot more sense. I thought you had a kangaroo there, but it makes a lot more sense. Thank you very much, Professor Lavigne. That was a wonderful presentation.